So the topic of today is going to be fluorescence nanoscopy. So continuing with the previous lecture, but focusing on sub 10 nanometer resolution and trying to approach real molecular scale resolution. So before I start, a few words about our institute. So the Center for Bio-Nanoscience Research, CBION, is in Buenos Aires, is a rather small institute established in 2012. We have at the moment 9 groups working on different areas of physical chemistry, addressing biological questions. In particular, our group is a small group. Here you have 2 snapshots of a few years back and a more recent picture that we managed to take after the pandemics. And so we are a small group of physicists, chemists, biologists, but we are lucky to have, and we're working on different areas. So very brief overview of our research topics are optical manipulation of colloidal nanoparticles, self-assembled nanophotonic devices, especially using DNA origami techniques. Nanoscale temperature measurements is something that has caught our interest since a few years, and fluorescence nanoscopy. That's the topic of today. And on this area, we develop new methods, some applications to neurobiology and to 3D chromatin organization. But today we are going to be talking about new methods of fluorescence nanoscopy delivering sub 10 nanometer resolution. So we are a small group, but we have a strong network of collaborators in Argentina and worldwide. And the experiments I will show you today were done in cooperation with the groups of Stefan Hell and Tom Jovin in Göttingen, Alfredo Cáceres, Nico Sain, Mariano Bisbal in Córdoba, Argentina, Guillermo Acuña at the University of Fribourg in Switzerland, Philip Tinefeld in Munich, Damián Refojo in Buenos Aires, and Sabrina Simoncelli in London. Okay, so during the talk, we will discuss 3 different methods. I will go over them rather quickly. So if you have further questions or more interest, you can check the related publications or ask me after during the question sessions, of course. Okay. So a bit of motivation, you had it already in the previous talk, but I need to review a few concepts to explain how these new methods work. So fluorescence microscopy, you know, is a great tool. It's actually the preferred tool to visualize biological systems because it provides a specific contrast and high sensitivity down to the single molecule level. So you can visualize, for example, cells like this baby neuron here and observe the morphology of the cells or how proteins are distributed in different compartments of the cell, like you can see here. But now, if you would like to get more detail, for example, making a zoom on this growth cone of this neuron, you will see that the image starts to get blurred. And that's not because the microscope isn't working properly, but it's really due to a physical limitation of the imaging process due to the diffraction limit of light. So we would like to see these small regions better. Luckily, already since several years, we have different methods of fluorescence nanoscopy available. There is a first generation of methods where you can distinguish basically 2 families, like coordinate targeted nanoscopy based on scanning microscopes, where STED is probably the most popular method, and also coordinate stochastic nanoscopy methods, usually based on a wide field microscope, where probably the most popular method is STORM. You can see here 2 examples of performance. In both cases, the resolution is increased considerably. So these methodologies are becoming the new standard as they become more and more widely available, because of course, if you can see more, there's no reason why not to do it. This is like having a high definition TV. You don't want to go back to your old TV unless you're forced to do it. So you see more, so there's no question about their advantages. And in fact, these methodologies have enabled the discovery of structures that were previously unknown. And a great example is this one from the lab of Jiaowei Zhuang. They showed in neurons, in axons and dendrites, they showed this periodic structure composed of actin, adenosine, and spectrin that nowadays is called the membrane-associated periodic skeleton of neurons. Here you see on the left an example of the first publication in 2013, obtained by Storm. And here on the right, you see another example obtained by Stead in our lab. And here you see, for example, a confocal image of this same region. So this structure is absolutely invisible in conventional microscopy. So there's no way you can tell that there is this regular structure here because the resolution is not enough. But now, when you have a super resolved microscope, you can clearly see that these proteins that in conventional microscopy appear to be uniformly distributed, they are not. In fact, they are organized in a regular structure. And of course, visualizing these structures is the first step towards understanding their function in biology. Okay, so with nanoscopy, we can see more, no doubt about it. We can see supramolecular protein structures that otherwise were not very visible. But still, the resolution of this first generation of methods is limited. So it's important to understand that there is not a fundamental limit to the resolution, but there is a practical limit. So in theory, these methods can achieve unlimited resolution, but in practice, and in particular in biological systems, they reach lateral resolutions in the order of 15 to 60 nanometers typically. And in the axial dimension, the resolution is 2 to 5-fold worse. So, like we did before with a conventional microscope, again, for example, what you see here on the left down, you see a network of microtubuli that are clearly better resolved by Stead. But still, we know that microtubuli are actually a supramolecular structure of tubuli, and they have a structure, a quasi-helicoidal structure, and we would like to see it, for example. So we would like to do further zoom into this image to try to see the structure of the microtubuli. And again, the image gets blurred and we cannot see it. And the reason for that is that, again, our resolution is limited. So we cannot detect the supramolecular structure of tubuli in this case, because it's a bit too small. So if we would like to see it, we would need an extra push in the resolution to reach the sub-10 nanometer region. That's the motivation. Why? Because the structural proteins that conform these structures have a size of about 10 nanometers or 5 nanometers. That's the size of structural proteins. So if we can reach the sub-10 nanometer regime in resolution, we will start seeing how these proteins organize really at the molecular scale. Okay, so nowadays there are methods that can reach this regime, and to explain them to you, I first need to go over again very quickly how fluorescence nanoscopy works in general. So you heard in the previous lecture that the main underlying mechanism is the on and off switching of fluorophores. That's the key for fluorescence nanoscopy. In the first family of methods, the coordinate stochastic methods, also called single molecule localization microscopy, SMLM, one uses a uniform illumination. So we illuminate our sample with uniform intensity. There is no information on the illumination in these methods. And then how do they work? Imagine you have these molecules here organized in this smiley face, like here. And we illuminate them, and they all emit fluorescence at the same time. If the distance between these fluorophores is smaller than the diffraction limit, then a diffraction limited image would look like this, just like a big blob of signal, where it's impossible to tell where each fluorophore is. But now imagine we can switch them off. We can switch them all off. And then in a subsequent instant in time, some of them, just a few of them, can be switched on. And these few fluorophores that are switched on are well separated in space. Then we will obtain signals like schematically shown here, signals that are diffraction limited, but whose center is placed exactly on the molecular position. So we can determine the center of the signal and assign it to a fluorophore in our sample. And then we can switch them off and switch on other fluorophores, and again determine their positions, and so on and so forth. So if we repeat this multiple times, we will end up with a list of positions of the fluorophores in our sample. And with those positions, we can reconstruct an image that is no longer limited by diffraction. It's actually only limited, the level of detail is only limited by the error with which these positions were determined. Under this mechanism, there are a number of different techniques. The most popular ones are storm, palm, and paint, but there are many more. Okay, so in this, you can see that in this technique, the information is all the information is retrieved from the detection, from the image of the single molecules. In practice, one has to record data like this. It's a video where molecules are switching on and off in different positions of the sample. And after analyzing a lot of frames and determining the position of each one of these signals, one can reconstruct an image. So we have to take each one of those images and determine its center. And that can be done with a precision that is inversely proportional to the square root of the number of total photons detected, as explained in the previous lecture. And then we can end up with an image like this. So this would be the wide field image if we put all the signals of the molecules together. And on the right, you would see the single molecule localization image that is constructed after analyzing each individual signal and determining its center and placing then a bright dot on each on a subpixel of this image. Okay. So localization precision basically relies on a fit to the measurement of the PSF of the microscope, and high precision requires a large number of photons. So here you see on this curve a rough estimation of the precision. So to keep in mind, if you want to reach 10 nanometer precision, you will need a few thousands of photons, basically. Okay, so that was one family of techniques. The second family is a directed family of techniques, or coordinate targeted nanoscopy. So in this case, again, we will switch on and off fluorophores, but in a different way. So imagine you have now this line of fluorophores, and the distance between them is again shorter than the diffraction limit. So if we illuminate them with a focused laser beam, it would look like that, more or less. And if we record this fluorescent signal from them, we will obtain this kind of signal. Rather continuous signal, we cannot tell where each molecule is. Now imagine that we have some methods, some mechanism to suppress fluorescence emission with light. If such a mechanism exists, then we can overlap with the excitation focus another focus of light, but in this case, instead of having a maximum at the center, it has a minimum in the center, and it has a maximum in the periphery. And this light, now schematically shown here in red, is suppressing fluorescence emission. Then, if we scan these two foci over the sample, we will record a signal that extends over smaller regions of space, because it cannot, the molecules on the periphery of the excitation focus cannot emit fluorescence. And if we can do this more dramatically, eventually we will start distinguishing the positions of each individual molecule. In other words, our resolution is improving and improving as we increase the laser intensity of the suppressing beam or depleting beam. Again, under this common mechanism, there are a number of techniques that have been developed, and the most popular one is stimulated emission microscopy, or STED microscopy, but there are many others. Here you see an example a conventional confocal microscopy of two proteins on the surface of this parasite, and on the right you see a super resolved image obtained by STED, and of course the amount of information you obtain is considerably higher. Important to know about this family of methods is that in this case it's kind of the opposite as before. Here all the information is injected into the problem by the illumination. So our illumination pattern is determining where we ask whether there is a molecule or not. And we move this pattern with a scanner over our sample. So ideally, if we would have zero background and a perfect off switching, a single detected photon would tell us if a molecule is present in that position. So we could switch on these two lasers, wait a certain amount of time, and then we could say with some statistical confidence, if we detect a photon, there is a molecule underneath. If we don't detect a photon, there is no molecule in that position. And then we move to the next position and so on and so forth. Of course, in practice, there's always background, and this off switching is never perfect. So again, we need a large number of photons to achieve a high resolution. And the question is, okay, why we cannot get more photons? Well, the reason is that all molecules undergo irreversible photochemistry when we drive them to emit fluorescence. It's a process called photobleaching. So this photobleaching, this irreversible photochemistry, is what really limits the resolution achievable with these methods. So there are basically, under these conditions, there are basically two types of solutions. So the one solution is get more photons. So find a way to get more photons. And this has been done. A great example of that is DNA paint, where the same position of the sample is interrogated with multiple fluorophores over time. So basically there is no limit anymore on the photons you can use. And the other solution is get more information from the few photons you have. So, and there are also ways to do that. And that's the topic of our lecture today. I will show you different ways to obtain more information using the few photons that one molecule can give. Okay, so the first way of doing that is with a new concept that we recently kind of grouped under one umbrella called single molecule localization with sequential structure illumination. This is basically combining the best of the two previous families. So we will use single molecule localization, but we will also use a structure illumination to inject information into the problem. The first implementation of such an imaging technique was PyFLUX a few years back, but now we will show you a common framework. I will start with that to show you that many other methods that have been published in the past, for example, for single molecule tracking, not for imaging, but for single molecule tracking, can be used under the same, can be understood under the same framework, and that enables us to think of actually new implementations in a very simple way. So orbital tracking, for example, was one of the first methods to track single molecules using structure illumination. It consists of orbiting a Gaussian focus around the molecule, and then from the signal of that molecule inside this orbit, you can determine where that molecule is. So this was a method first proposed theoretically by Jörg Enderlein and first demonstrated experimentally in the group of Enrico Graton. Very recently, an apparently totally unrelated technique called MINSTED was shown by the group of Stefan Hell, but we can think of MINSTED as an orbital tracking experiment using a subdiffraction effective PSF. PyFLUX, PyFLUX is a different technique in the sense that it uses a focus light with a central zero of intensity. And it uses, again, this, in this case, it's not a Gaussian beam, it's a beam that we call a donut focus over several positions to determine the position of a molecule. And single molecule confocal localization, basically it consists of obtaining an image of a single molecule by raster scanning a Gaussian focus over a molecule, and in this way acquiring an image of that molecule and using that image to determine its position. This has been shown by the group of Enderlein recently. But all these methods, they have a basic common components. So basically, all you need is an excitation field that depends on position. So the illumination must not be uniform. It has to be some shape. It could be a Gaussian, it could be a donut, it could be something else, but the intensity has to depend on position. That's the key component. And then we need to shift that illumination over different positions of the sample, and we have to record the intensity of a molecule on those different exposures to the excitation field. And by doing that, we will record a different number of fluorescence photons. So if we have K exposures, for example, to the excitation field, we will record K number of photons here, N1, N2, N3, Nk, number of photons, each one to the different exposure. And from this set of detected photon numbers, we can calculate what is the most probable position for that molecule. And to do that, we use the maximum likelihood estimation process that is a standard, and it's basically computing the multinomial probability of this different photon detected photon numbers, considering that fluorescence is linear with the excitation field, which is normal, usual conditions. Okay, so that's all you need. And now you can, with this framework, you can describe orbital tracking, you can describe mean state, you can describe mean flux, you can describe single molecule confocal detection, or you can describe new ideas, for example, of doing a raster scanning image using a donut field, or doing orbital tracking with a donut beam that we call here RASTMIN, or any other thing that you could come up with. You can describe it with this framework, and this is useful to compare the different methods in a fair way and see which one is more efficient in different aspects. Here you see, for example, a calculation of, for orbital tracking. Orbital tracking using a Gaussian beam has a characteristic that it has an optimum radius for the orbit that is roughly half of the full width half maximum of the excitation field. So you can, if you use a smaller radius, the method becomes worse, and if you use a larger one, the method becomes worse again. So there's an optimum radius. And here you see results for such a orbit, and you can see that in this case it's using 500 photons. You can get very high precisions in the order of 10 to 12 nanometers inside the orbit. Here you see
no, no está todo cortado, es como que Hi, Fernando, can you hear us?
Hi, can you hear me now?
Yes, we had like we got se cortó.
Yes. Something happened. Yeah.
Okay.
I'm gonna share a screen again.
Great.
Sorry. Okay.
No problem.
Short break so everybody wakes up. All right, so orbital tracking can be described by this formalism. The same happens with orbital tracking using a minimum of light, which is very interesting because now there is no limit, there is no optimum radius. You can make it as small as you want. And as you make it smaller, you will find that your precision becomes better and better using the same number of photons. MinFlux is a interesting configuration because it's kind of the minimum raster scanning configuration, because you have like 3 exposures that define an orbit and 1 exposure in the center that is like is used to probe the inner part of the orbit. So it's a smaller image you can think of. And it's very efficient. It's actually one of the most efficient. And this one, we found it particularly interesting because it's raster scanning a minimum of light. And you see that the localization precision here on this map is really, really good over a large area on that raster of minimum. Okay, so here you see a comparison of all the methods. And what we could learn doing this is that all methods using a minimum of light, like MinFlux, RASTMIN, OTMIN, these are the blue and light blue curves here, are better than the methods using a maximum of light, considerably better. The red curve here is MinSTED, which is also very good, but of course is using a sub-diffraction effective PSF. All the other methods are using diffraction limited foci and are therefore much easier to implement. Okay, so here you see an example of MinFlux in practice. So you need to have a donut-shaped focus and switch its position between these 4 positions and record the fluorescence emitted by a molecule. And in each position, you will record a different number of photons from which you can determine the molecular position. So here you see, well, what you see here is a comparison of the localization precision of MinFlux in comparison to ideal cameras or cameras with very high signal to background ratios. And you see MinFlux is always better, but you also see what I told you before, that when you use minimal light, you can reduce your field of view to make it as small as you want, and the precision becomes better and better. For example, here the blue curve is MinFlux using a field of view of 150 nanometers. And you see that, for example, with 500 photons, you get a localization precision of 3 nanometers. If that's not enough, you can reduce the field of view, for example, to 50 nanometers. And then your localization precision is about 1 nanometer, using the same number of photons. So for a given photon budget, you can become increasingly better in the localization by making the field of view smaller and smaller. So you can use MinFlux or any other methods of this kind to do nanoscopy in a small area by combining it with single molecule blinking, or you can do nanometer tracking inside this small area by following a molecule position in time, or you can do tracking over larger distances if you have ways to recenter periodically your excitation pattern, okay? Here you see an example of MinFlux to obtain nanoscopic images. Here we used as a calibration standard a DNA origami structure where fluorophores were organized at distances of about 10 nanometers or even short of 6 to 3 nanometers. And you can see that MinFlux can clearly resolve them. The scale bar here is 10 nanometers now, and we can clearly resolve these structures using, in this case, 500 photons, and here about 1,000 photons. These are values that are easily achievable with standard fluorophores. You can also do tracking, and MinFlux has also important advantages for tracking, because since it uses a very few photons to obtain high localization, you can play with that. And for example, in this case, in this tracking example, we were happy with the localization precision of about 50 nanometers. Not so great, but it's really a good localization precision for tracking. But the point is that 50 nanometer precision with MinFlux, you can obtain it using only 9 photons. So detecting 9 photons, you can get this localization precision. And detecting 9 photons is also very fast. You don't need too much time to collect 9 photons. So we could get 50 nanometer localization precision with 125 microseconds time resolution. So in tracking is always important this interplay between time and spatial resolution, right? So here you see this is useful, for example, to obtain a diffusion coefficient of these proteins at video rate. What you see here is a diffusion coefficient with a 35 millisecond sliding average. So you can see with video rate how this protein is diffused with a certain speed, then they get stuck in some position, and of course you know where they got stuck, then they start diffusing faster, then they get stuck again, and so on and so forth. This kind of data is not possible to obtain with a camera-based tracking, for example. More recently, we showed a new implementation of MinFlux that is called pulsed MinFlux or p-MinFlux, and it uses pulsed interleaved excitation to do the same localization scheme, but in this case, we do it very, very fast. We do it at the repetition rate of the laser at megahertz. So this is the fastest way of doing MinFlux, and at the same time, it gives you access to lifetime information. So I will show you an example next. Before I want to tell you that all this implementation of MinFlux is open source, written in Python, and you can get it from the GitHub of our group if you are interested in. And here you see examples. Again, we use the DNA origami to test the performance of the methods. And here you see an image now with a 5 nanometer scale bar here. And at the same time, you can obtain lifetime because it comes for free on these methods. So I think this is one of the few, or probably the only super resolution technique that provides clean access to the lifetime, because here we are not modifying the lifetime of the molecules, and we can therefore use it to detect changes in lifetime and ascribe them to changes in the environment or molecular conformations, etc. Or here you see another example using a dynamic DNA origami to test the lifetime and time resolution performance of pulsed MinFlux. Well, and even more recently, we implemented this RASTMIN method, which is a related method to MinFlux because it uses also a minimum of excitation light, but it's much easier to implement because you can implement it on any confocal microscope, because all you have to do is to obtain a raster scanning image of a molecule. And basically you need to do this twice. You need to do it once to obtain a reference measurement. This measurement, you can do it many times and average it, and in order to obtain a very precise measurement of your PSF. And then you can perform a single molecule measurement, which is of course a low signal to background ratio and a lower and with higher noise, but using the information from the reference measurement and the positions, you can obtain the position of that molecule with high precision, comparable and even a bit better than MinFlux. Here you see examples of performance. This is localizing a molecule with only 250 photons. The localization error is 3 nanometers. With 1,000 photons, the localization error is already 1.4 nanometers. And with 2,000 photons, the localization error is 1 nanometer. And here you again, you see an image of a DNA origami with distances between molecules of 20 and 15 nanometers, which are fully resolved. Okay. Next idea to reach sub 10 nanometer resolution is called simpler. And again here, we will inject information with the illumination, but we will add a new component here. This method it works under TIRF conditions. So you know when you do TIRF microscopy, you illuminate your sample with angles of incidence higher than the total internal reflection angle, and therefore the illumination light doesn't propagate inside the sample. Instead, you have a decaying intensity. So your illumination decays exponentially into the sample. This is well known, and this is the key phenomenon that gives TIRF microscopy this surface sensitivity. But also, in addition to this, individual molecules that are close to this interface also emit light in a different way, depending on the distance to the interface. And what happens is that the closer they are to the interface, the more light they will emit into your microscope objective. Here you see calculations for a molecule that is perpendicular to the water glass interface, and here for a molecule that is horizontal, parallel to the water glass interface. In both cases, the closer they are to the interface, the more light they emit into the glass and therefore into our objective. So you have two effects that make molecules brighter if they are closer to the glass interface. So if you compute these two effects properly, you can obtain a calibration of your signal depending on the distance to the interface. And that's what we did here. We calibrated this properly, and you can find the details on this paper. And then if you have a proper calibration, which is easy to obtain, then you can combine this method with any single molecule localization method. Remember, in single molecule localization, what you do is you detect single molecule events, like this one here. A molecule switches on, and after a few frames, it switches off. And from these signals, you determine the position, the center position of this signal, remember? And that you collect it to produce a super resolved image. So what you have to do now is, on top of that, in addition, you have to take the frames that where the molecule was fully active, fully emitting the whole time, like this, in this case, frame number 3 and frame number 4, and then you can compute the average intensity of the molecule. And from that average intensity, usually represented by a number of photons, and you can determine the axial position set. It's very simple, and it works very nicely. You can see here an example of performance. Here you see microtubuli in 2D, super resolved, but now applying simpler, we can obtain cross sections of the microtubuli, like this 4 examples you can see here. And these structures are very small. They have a diameter of 40 nanometers. And we can clearly resolve their cross-section in the axial direction. Okay, again, if you're interested, you can find an open source Python software to implement simpler in your lab. It's actually not hard. You can download it from our GitHub. Well, and finally, and I have only 5 minutes or less to tell you this, is another new technique called STED-FRET. And as the name says it, it's a combination of FRET and STED. And probably I don't need to spend time explaining FRET to you. It's Förster resonance energy transfer. It's a widely used phenomenon to study molecular interactions and molecular environments using fluorescence signals. So it's widely used. There are numerous protocols to implement FRET imaging, but of course, diffraction limited FRET imaging has limitations because we are only obtaining an average information over many FRET pairs inside a large volume, and also so that we don't know exactly where those interactions are taking place. So it's interesting to try to obtain a super resolved FRET pipeline. And we have achieved this using STED. The setup consists basically of a 2-color STED microscope, nothing more. And all you need is, okay, here is the examples using DNA origami. I have to go quickly over this. We can clearly resolve the 2 FRET modules that we built on this DNA origami. And we can also see them performing dynamic FRET, and we can super resolve each one of the FRET modules. So then all you need is a pipeline to compute FRET signals from STED images. And we have done that using a formalism developed by Tom Jobin and Ellie Harris a few years back. It's an intensity-based FRET measurement that is suitable for STED images. So the only extra care you have to take is that since we are not averaging, like in diffraction limited FRET, you will find variations, local variations of concentrations of donor and acceptor molecules, because the volume we are interrogating is smaller and smaller. And therefore, you will have fluctuations of apparent FRET signals simply because maybe we have fewer donor molecules or fewer acceptor molecules. And you need to take that into account. We can do that by performing a control measurement. That is this histogram here. This is the control measurement of a sample with no FRET. And you see that we detect values that are non-zero, of course, due to these fluctuations. But in a sample where FRET is occurring, you see here in blue, the distribution of FRET values is very different. And in confocal, these two are very well separated. But in state FRET, these two are not totally separated because, as I said, the diffraction, the detection volume is very small. And therefore, there might be random fluctuations that give you an apparent FRET signal. So you need to put a threshold that statistically will have some false negatives and some false positives, but you can compute that with your control measurements, and putting that threshold properly, you can know that you will only consider positive FRET signals in diffraction limited volumes. And this is what we did here using the membrane associated skeleton of neurons as example, and we could detect molecular interactions in subdiffraction regions of the sample. And this is something that it's impossible to obtain by super resolution alone or by FRET measurements alone. You need to combine the two. Okay, with that, I finish. And of course, I understand that not everything was totally clear. So if you have any questions, I'm happy to answer them now.